The overall goal of this group of experiments is to identify developmental defects in the nematode Caenorhabditis elegans during the early and late phases of embryonic elongation.
This method can help answer key questions in the developmental genetic field. It enables the characterization of genes controlling a late stage of the Caenorhabditis elegans embryonic development called embryonic elongation. The main advantage of this group of techniques is that they identify genes controlling either early or late stage of the elongation and also those that control morphogenic processes at the anterior or posterior part of the embryo. I will demonstrate the procedure together with Olivier Rocheleau-Leclair, a former student of the laboratory.
This procedure requires DIC lenses, prism, and a video capture system for time-lapse microscopy of Z-stacks. First, position the slide-mounted sample for viewing. Preparation details are in the text protocol. Beginning at low magnification, identify embryos in their pre-morphogenesis stages. Then, add a drop of oil and use a more powerful objective. Now, identify the top and bottom points of the embryos to record. And set the Z-stack parameters. Set the time-lapse to take images every 2 minutes. Make certain that the light shutter is closed between the image acquisitions. The time needed to capture the early elongation event can vary from 1 to 8 hours, depending on the stage of the embryo. Now, run the image acquisition program.